Good morning friends, I welcome you my channel Educational Information for All. I request you to subscribe my channel and hit the bell icon for new video notification. I start Neet Biology Chapter 2 Biological Classification question number 28. Curing of a tea leaves is brought about by the activity of four options they given: fungi, bacteria, mycorrhiza, viruses. Answer is B. Curing is a process done to add species flavor and taste in the tea leaves. It is also done for tobacco. In this process, after harvesting, the curd leaves are hung in shade and are permitted for the action of bacteria. The curing of tea leaves is done by a Micrococcus candidans. Micrococcus is a gram-positive aerobic bacteria which is a member of Mycococcaceae. Next, we see question number 29. All the following statements concerning the Actinomycetes filamentous soil bacterium Frankia are correct, except that Frankia options can induce root nodules on many plant species. B. Can fix nitrogen in the free living state. C. Cannot fix specialized vesicles in which the nitrogenous is protected from the oxygen by a chemical barrier involving tryptophan hopenoids. D. Like rhizobium, it is usually infects its host plant through root hair deformation and stimulates cell proliferation in the husk cortex. Answer is B. Frankia is a nitrogen fixing symbiotic bacteria. It induces root nodules just like rhizobium. It is associated symbiotically with the root nodules of several non-legume plants like Cassarina, Alnus, rubus, etc. It cannot fix nitrogen in free state. Question number 30. For retting of jute, the fermenting microbe used is four options: methanophilic bacteria, butyric acid bacteria, Helicobacter pylori, Streptococcus lactin. Answer is B. Butyric acid bacteria. Retting is the process of separating fibers that are held together in close association using a variety of bacteria. Fibers of jute are held together in close association and they are separated by the action of butyric acid bacteria. Example: Clostridium butyricum. These plants are immersed in water so that they absorb water and swell. Due to the activity of bacteria, the pectic substance of middle lamellae are hydrolyzed and the fibers are separated. These separated fibers are used in making up ropes and sacks. Question number 31. Mesophilic prokaryotes, four options: grow and multiply in very deep marine sediments. B. Occur in water containing high concentrations of bacterium hydroxide. C. Readily grow and divide in seawater enriched in any soluble salt of barium. D. Grow slowly in highly alkaline frozen lakes at high altitudes. Answer is A. Mesophilic prokaryotes are facultatively anaerobic bacteria. They grow and multiply in very deep marine sediments. Most Basophilus grow better at pH of 8.5 or high. Next, 32. Which statement is correct for bacterial transduction? A first, transfer of some genes from one bacteria to another bacteria through virus. B. Transfer of genes from one bacteria to another bacteria by conjugation. Bacteria obtained its DNA directly from mother cells. D. Bacteria obtained DNA from other external source. Answer is A. That is transfer of some genes from one bacteria to another bacteria through virus. In transduction, genetic material of one bacterial cell goes to other bacterial cell by agency of bacteriophages or phages, that is virus infecting bacteria. Transduction was first of all reported in Salmonella typhimurium by Gender and Ledberg in 1952. Transduction is used for gene mapping and analysis in bacteria and also for stain construction. Next 33. In bacteria, plasmid is A. Extra chromosomal material, B. Main DNA, C. Non-functional DNA, D. Repetitive gene. Answer is A. Extra chromosomal material. In addition to the nucleoid, bacterial cytoplasm normally contains many small separate pieces of DNA called plasmids. Next 34. Organisms which obtain energy by the oxidation of reduced inorganic compounds are called photoautotrophs, chemoautotrophs, saprosic, coproheterotrophs. Answer is B, that is chemoautotrophs are organisms that are capable of manufacturing their organic food utilizing chemical energy released in oxidation of some inorganic substances. The process of manufacture of food in such organisms called chemosynthesis. 35. What is the true for archaebacteria? A. All halophytes. B. All photosynthesis. C. All fossils. D. Oldest living beings. Answer is D. Oldest living beings. Archaebacteria are believed to have originated at a time when there were extreme conditions in the bisosphere. Bisosphere. Sorry. 36. Difference in gram-positive and gram-negative bacteria is due to cell wall, cell membrane, ribosome, cytoplasm. Answer is A. Cell wall. If retain the stain after washing using alcohol is called as gram-positive. If not retain the stain after washing using alcohol is called gram-negative bacteria. 37. Transfer of genetic information from one bacterium to another in the transduction process is through bacteriophages released from the donor bacterial stain, another bacterium having special organ for conjugation, physical contact between donor and recipient strains, conjugation between opposite strain bacterium. Answer is A. Bacteriophages released from the donor bacterial stain. The explanation given in question number 32. Next 38. Bacterium divides every 35 minutes. If a culture containing 10 raised to 5 cells per ml is grown for 175 minutes, what will be the cell concentration per ml after 175 minutes? The four options they given, you want to do a calculations. Answer is B, that is 32 into 10 raised to 5 cell. I given the calculation, please see it. Take a screenshot. Next 39. The DNA of E. coli is Escherichia coli. A. Double stranded and linear, B. Double stranded and circular, C. Single stranded one and linear, D. Single stranded and circular. Answer is B. Double stranded and circular. E. coli is a gram-negative rod-shaped motile or non-motile bacteria. E. coli contains a double-stranded DNA as its genetic material. The DNA is not associated with any histone proteins, so it is referred to as naked DNA. This DNA is circular with no free ends. Next 40. The main role of bacteria in the carbon cycle involves chemosynthesis, B. digestion or breakdown of organic compounds. C. Photosynthesis. D. Assimilation of nitrogenous compounds. Answer is B. Bacteria are responsible for maintaining the conditions of life as Earth by virtue of their powers of decomposition of plant and animal bodies, by which the limited supply of CO2 available for photosynthesis is re- replenished. Thus, they act as decomposers in the carbon cycle. Next, 41. A few organisms are known to grow and multiply at temperatures of 100 to 105 degree Celsius. They belong to A. Thermophilic sulfur bacteria, B. Hot spring blue-green algae, C. Methanogenic archaebacteria, D. Marine archaebacteria. Answer is A. Thermophilic sulfur bacteria. These are thermoacidophiles, temperature and acid loving, you can say. Archaebacteria are found in hot sulfur springs. 42. The heredity material present in bacterium E. coli is 4 options: single stranded DNA, double stranded DNA, DNA and RNA. Answer is B. Double stranded DNA. The explanation given in question number 39. Next, 43 we see Azobacter and Bacillus polymaxa are the examples of A option pathogenic bacteria, B decomposer, C symbiotic into fixer, D non-symbiotic nitrogen fixer. Answer is D non-symbiotic nitrogen fixer. Azobacter is a free-living bacteria which occurs in soil and fixes nitrogen directly. Bacillus is also free-living bacteria which act upon nitrogenous excretion and protein of dead bodies of living organisms. Next, 44. What are the sex organs provided in some bacteria? A. Sex pili, B. Plasmid, C. Circular DNA, D. Gametes. Answer is A. Sex pili are minute and non-flagellar hair-like structures projecting from the wall of many gram-negative bacteria and few gram-positive ones. They are entirely composed of a protein called pillin. Next, 45. Which type of DNA is found in bacteria? Circular free DNA, membrane bound DNA, straight DNA, helical DNA. Answer is A. Circular free DNA. Bacterial cells do not have nucleus. Nuclear material of bacteria lies free in the cell in the form of an irregular, thin, fibrillar and circular single molecule of DNA called nucleoid or chromatin body. 46. A large number of organic compounds can be decomposed by Azotobacter, chemolithotropes, mycoplasma, Pseudomonas. Answer is B. Chemolithotropes. Chemolithotropes can derive the energy required for growth from the oxidation of inorganic compounds. Thank you.